Welcome back, everyone. I hope you're all energized after the tea break. So I would like to invite Dr. Sanjeev Kumar, President of Biologics R&D at Zydus Life Sciences, to moderate the following two sessions. I would also like to invite our next speaker, Professor Dipankar Nandi. Please give him a huge round of applause. Sir, I request you to introduce Professor Nandi.
Hi, welcome back everybody. So we have now Professor Dipankar Nandi, who is a professor in the Department of Biochemistry at the Indian Institute of Science, Bangalore, and who possesses tremendous expertise in immunology, specifically focusing his work on studying novel microbial genes during stress and infection and understanding host-encoded immune responses. He holds a PhD from the University of California, Berkeley, and has completed postdoc earlier at the Department of Molecular Genetics at the University of Cincinnati, USA. And he has been a professor since 2012. His work delves into various aspects of immunology, such as investigations into host responses in fast-deficient autoimmune-prone LPR mice when infected with Salmonella typhimurium. Okay. Immunomodulatory effects of a plant-based sesquiterpene lactone and inhibiting immune cell responses, and functional characterization of Salmonella typhimurium encoded cold shock protein E during stress responses, biofilm formation and virulence. He also holds a patent, and we welcome Dr. Dipankar Nandi to give his talk.
All right, okay. Thank you so much for the kind introduction, sir. Let me start off by thanking the organizers for this invitation. And what I will be talking about is on antigens. When I was first asked to speak on it, I thought I was wondering, what to speak on. But anyway, I've sort of tried to put together a talk. So this is the first time I'm going to give a talk exclusively on antigens. Anyway, let's start off. So this is the Indian Institute of Science, and this is Jamshedji Tata. Some of you might be familiar with this. And right now, actually, the flower show is on, and it's an annual event where the horticulture department puts up a flower show in honor of the upcoming birthday of Jamshedji Tata, whose vision as well as whose generosity started off the institute. Apart from Jamshedji Tata, the land was given by the Maharaja of Mysore, and the British government, oversaw the creation of the institute, which is more than 100 years old. So the department, this is the Department of Biochemistry from where I come from, and this was started in 1921. It was one of the oldest departments of biochemistry in this part of the world. And we were here till 2011 end. We moved to this biological sciences building, which houses other departments also, and we did this end of 2011. We started off in about 2012 over here. So my laboratory works on different aspects, and which is shown in a form of a collage which was made by a student over here. But I will not be speaking about any aspect of my laboratory's work for this meeting today. And I will treat this as exclusively as a teaching module, and I will also treat the lecture as a meal course. So we'll have appetizers, and since I'm the first speaker over here, there'll be appetizers, there'll be the main course, and there will be a dessert. So that's how I've structured it. So the appetizer part why study immunology? This becomes important. The way I also do it is as an interaction. So I would be asking for a feedback. So the interaction is important because it ensures that you are not sleeping during the talk. And I usually, for my teaching lectures, that's what I usually do. So I will try doing that over here, but keeping the aspect about time also. The main course will be on antigens. I will discuss antigens. I will discuss both B cell antigens as well as T cell antigens, and you will see why that is important. We'll end with some highlights. Okay, so why study immunology? So as I said, I want this to start off at least as an interaction and not as a one-way affair where I speak and you all listen. So why study immunology? I thought this was going to be, for people who are not all that familiar. So why study immunology? You can put your hand up, and then we can have a small discussion on this. So the other thing that I tell my students is not to memorize, because if you memorize, once the exam is over, that neuron is going to die. So it's best not to memorize, but to look around. You can just look around your own family, your friends. You can do some reading material. You can read up. I mean, you have lots of opportunities these days. Kids spend a lot of time on their mobiles. So you can do some useful exercises also. So why study immunology? Yeah, go ahead.
Whether we study it or not, our immune system will go on working and will defend our body. But sir, knowing about immunology makes us conscious of that defense system first, and also we can add upon and make use of that knowledge in order to defend ourselves.
Very good. Very good. Okay, so she's given a very good answer, but I'd like some more concrete examples of why we should study immunology. And give some concrete examples. Don't. It shouldn't be all up in the air. Give examples. Something that you have seen, something that you have read up. I mean, as far as history goes, we have recently had some incidences which I think should jolt your memory a little bit. Even though public memory is short. Go ahead, don't worry. It shouldn't be only one student is answering, there should be, as I said, don't be afraid. I mean, there's nothing, what is there to be afraid of? There's nothing to be embarrassed about. Yeah, go ahead.
As Professor Madan said, to keep ourselves away from disease, we should study immunology.
True. Give some examples. Give concrete examples. Yeah, go ahead.
Total eradication of polio, that has been a very big achievement for our country.
Yeah, very good. Yeah, so she says, removal or eradication of diseases, smallpox, polio. She mentions polio. I will also rake up polio. But something in your family, something if you look around, somebody with some problem. Yeah, go ahead. Yeah, that's good. Okay, so it's slowly beginning to start. That's good. Yeah, go ahead.
As we say, precautions are better than cure. So, like we can see right now, the allergy season is going on. Pollen allergy is there.
Yeah.
If we know about hypersensitivity and all, we can prevent from pollens and also that.
Very good. Excellent. So she's talking about allergens, and the problem, and the one that she picked on because summer's starting, the birds are, there's a lot of flowers, so there's pollen allergy and so on. Very good. That's a very good example. So exactly some, like that, can you think of some examples that you see? Yeah, go ahead, yeah.
Organ transplant.
Yeah, organ transplants, fine. We will cover organ transplant. That's good. Okay, go ahead.
Yeah, so in general, we need to, first question is why not? Second is that because of a lot of innovations are based on that. I recently read a research paper which was on using some photosynthetic microbes to provide oxygen in the hypoxic conditions that are developed in the brain. We would not have been able to do that if we would not be able to nullify its effect on our body and nullify the immune response it is causing us.
Okay.
So to have.
All right. Okay. Yeah. So any other examples? As I said, look around in your family. I think that would be. Yeah. Go ahead. Yeah.
Sir, some home remedies like we use in COVID-19 times. So
no. What I meant was, something that you see in your family members, and that makes you think that maybe it's important to study, or your friends for that matter. I mean, something that was said was, organ transplants, those are important, skin transplants. Other thing was pollen allergies, something like that. Yeah, go ahead.
So, sir, these like many diseases like cancer, autoimmune diseases, multiple sclerosis.
Yeah. So, autoimmune diseases, is something very common, and it sort of tells us that we should be at least be aware of these aspects. Any other?
a dust allergy?
Yeah, allergens. So all those would be a part of it. Okay, fine. So I think we've done a reasonable review. So so I have a bunch listed over here: allergens, asthma, all right, autoimmune diseases. Lots of it. If you just look around, there are people, in our families who suffer from autoimmunity. Women, for example, are much more prone to autoimmune diseases than men. Men are more prone for cancers and so on. Right. So I mean, you will see this. Boost response to pathogens. We talked about vaccines, right? A major game changer that's increased our lifespan. As she mentioned, polio is a big thing, but polio is still prevalent in two countries, Afghanistan and Pakistan. So why is it that even though that there is, that there are vaccines for it, why is it that some countries, you still have it? So then immunodeficiency, we will talk a little bit about it. Organ transplantation, there's a lot more, but just to get us thinking about it and getting us acquainted with this. So this is a X-ray of a hand deformed by rheumatoid arthritis, and which is a major problem. And that's why I said you don't have to memorize, just look around, right, and you will be able to see. This is progress of a HIV, untreated HIV infection. You can see that the blue line is the AIDS virus, so it comes in over here. The immune system, tries to suppress it, and so it goes down. And then you have variants and so on, and then it comes up and sort of takes over. And as you progress over here, the person comes down with AIDS. You come down with AIDS because your CD4 T cell counts are going low, and at some point they are so low that the virus takes over. So which is a major problem. So as Taruna mentioned about immunity, what is immunity? And this comes from exemption. You're exempt, right, from something. So often what happened is people would go to fight as soldiers, and because they were putting their lives on line, they were immune from paying taxes. And then people found out that those people who suffered from infections were also immune. So in this case, plague and so on. So even before the smallpox vaccine was discovered, people knew that, especially this started off in Turkey and then India and China, this was known. Smallpox has a major mortality rate. It's about 30%, right? But people realized that if you took the dried pustules over there and then inoculated, you would get a mild form of disease, but you were protected, right? But this was all empirical, but it led to the development of what's known as tikka and tikkadar. So you had a group of people who would be giving these things, but it was dangerous because it was not standardized. So if too much of the pustules were given, some people would come down with the disease and may even die, right? So I mean, until the vaccines came over, it was not really standardized as much, but you did have this aspect, and that's how the name tikka and tikkadas come from. Interestingly, when the vaccines first came in, the group that opposed the advent of vaccines were the tikkadas, because they were afraid that they would lose their business, right? So until the government sort of got them to be a part of the vaccine effort, and since then, it's been a success. So it's very interesting. So immunology is linked to history, so don't disregard history. So which is also important. So the other aspect that I'd like to discuss is, why did the immune system evolve? As a lot of you mentioned, it's to help protect us, and which is important. So over here, might be interesting to think in terms of doubling time, right? So what is the doubling time of, I mean, this is a typical question. What is the doubling time of, let's say, a bacteria, let's say E. coli? Yeah, I knew everyone would jump at that. Okay, now I'm going to ask you, what is your doubling time?
24 hours.
24 hours.
Your doubling time is 24 hours? Do you double that fast? I mean, our population is quite high. Imagine if you doubled at, at 24 hours. So what's your doubling time? So E. coli doubling, you've learned, and the whole class almost erupted at about 20 minutes, and that's quite straightforward. But your doubling time? Yeah, go ahead.
Sir, because we are
time on what cell you're talking about. Like,
I said your doubling time, our doubling time, human doubling time. So complicated. When did your parents have you? About 25, around 25, 30 years, right? So your doubling time or our doubling time is about 25 years, 25 to 30, right? Depending on when your parents had you, right? Doubling time of E. coli is 20 minutes. So you can clearly see we are at a disadvantage. We need help, right? So, I mean, so this sort of tells you, if you think in terms of doubling time, it's a good way for you to think of, why we need help. We need protection, right? So it's very important that you keep that in mind when you're thinking about immunology, why you need help. This is a good way for you to think in terms of it. Okay, so the other way to think about it is to think of the immune system as part of a sort of a defense network. And again, most of you might be aware, in states that are not disturbed, you have what's the BSF. They are the first line, right? So if there is a breach, then the BSF calls for help with regard to the army. And getting the army to help also is not straightforward. You have to contact Delhi. Delhi has to get its act together, then send the army. It takes some time. But the immune system is pretty much that way. So we have, troops that are patrolling, but they're partly, they're border security force, right? And if there is a breach, then they call for help, and then the armed forces comes in. When the armed forces comes in, you have the armed forces as, Army, Air Force, Navy. You have different aspects, and even within each, there are subdivisions. So you have the infantry, tank, paratroopers, artillery, and why have it so complicated? Why not just have the army, or why not just have the Air Force and just have a single set, maybe just Jawans? Why have all these different sets? I mean, and you will see that the immune system is also complicated. It's just not straightforward. They have just so many different types of cells, so many types of molecules, and so on, right? So why have so much complexity? Yeah, go ahead.
to protect us from different
No, I mean, as far as protection, you could just have one type, and that would still do the job of protection, right?
The mechanism of action of every action is different. So if someone
I think what she's trying to say is the enemies, there's not a single form of enemy, right? So the enemy keeps changing, right? And so therefore, if the enemy changes, we need to have different ways of protecting us, right? So even with the recent virus, you have different variants, and why do you have different variants? The virus is basically trying to look after itself and trying to find out ways to propagate itself, right? So that's why it changes itself in the hope that, some form will sort of propagate itself much better. So in order to do that, we need to have, and the fact that we have evolved for so many years and been successful, it's because we have had a mechanism to cope up with all these variations, right? And different forms that may come up, different forms of enemies and so on. So that's, something, to keep in mind. And that's why the immune system has developed this way. So do keep that aspect in mind. So now I'll give two examples on why the immune system is important. The first is, you must have seen this with AIDS patients, for example. What happens if you have a compromise in one part of your immune system? So for example, your CD4 T cell population goes down. Once that population goes down, you become severely immunocompromised. But it's only one cell population that's going down. So why do people become so severely immunocompromised when one cell population goes down?
because one cell is actually leading to activation of much more other cells also.
Yes, so actually it's just not about one cell, it's about different nodes, and some nodes play more important roles than others, right? And with the CD4 population, as she said, they are the helper populations. They are the helper populations because they help others, right? They help B cell, they help macrophages, they help many different types of cells, and that's why it's important. So if that population goes down, the entire immune system gets compromised, as a result of which you have these patients being severely immunocompromised, and that's why they suffer from so many different types of infections. So the other example that I'll give you is, is a very well-known example, that of the bubble boy. And the bubble boy was David Vetter. And there are books written on David Vetter. There's been a movie made on it. So this person lived in a bubble, right? And again, because of the pandemic, most of you might be sort of familiar with this. And this was, also important because this was actually an experiment that was funded by the NIH, and it was funded for several years, right? And so over here, this is the boy David Vetter. They knew that he would have X-linked disease, as a result of which, from the time of his birth, he was put in a bubble, right? And as I said, and this was on for several years. The baby became big, and as he became, bigger, right? There were, other problems that were coming up. With respect to funding, with respect to doctors, taking care of him. Also the fact that, a lot of us take interactions for granted. And if we don't have those interactions, it affects us in many ways. So we see what others are doing, we have peer interactions, we learn a lot. So because of that, what they thought, now his sister was just fine. So, so they thought that they could take the bone marrow of the sister, put it into him, and sort of repopulate the immune system. And unfortunately, that experiment, did not work well because the sister's, bone marrow had a hidden virus, and at that time they had not screened for it. So unfortunately, when the bone marrow was put in, the virus exploded over here, and the poor kid died. But it tells you of the important role of our immune system. So the sister had the virus but was still fine. This poor kid was immunocompromised, as a result of which the virus took over. He had to be protected, and if that protection was not there, he unfortunately passed away. So it also tells us, how much, we need to, give respect to our immune system, and the fact that we are here, all of us are here, and, we really need to appreciate the immune system for this part, right? So there are two main parts. One is the innate. Innate is something that we are born with. Some people are innate singers, innate musicians, innate artists, and so on. So this is the initial response. They are the body's BSF. They are responsible for the initial response. They recognize invasion, limit damage, and they call for help when required. The other is the adaptive. Now, the adaptive takes some time to sort of come in because the troops arrive, but there are two important aspects about the adaptive response. One is they are specific. They are very targeted. They know which variant and so on, right? The other aspect is memory. They have memory. In fact, the basis of all vaccines is the fact that we have memory. So that's something that's important for all of us, because you take vaccines, and in case you come across the pathogen at a later point in time, you're still protected with it. All right, okay. So again, you have different cells over here. You have the innate cells over here, adaptive. I will be focusing a little bit on this, but we will see this a little bit later. Okay, now B cells and T cells recognize antigens in very different ways, and again, this is something very important for all of us to realize. So these are three different clones of B cell. However, only one clone gets to bind this antigen, right? As a result of which this makes more of itself, right? Makes more clones, and then some remain as a pool of memory cells. The others, secrete these. These are plasma cells that secrete these antibodies, as a result of which the antibodies are out in circulation. That's why antibodies are part of what's known as humoral immunity, which means you can take them away from the cells and they are still active. In fact, the basis of plasma therapy, again something that you might have heard of during COVID, was this, that the antibodies could be taken from the plasma and they could be given, and they may be protective. In some cases they have been protective, but but that's something to keep in mind. So this is structure of this is a membrane-bound antibody molecule. This is the heavy chain, light chain, and then you have the antigen binding parts over here. T cells, on the other hand, recognize antigens in very different ways, right? So this is a T cell, and again, T cells are specific. Specificity comes from the T cell receptor, and they see their antigens in context of the MHC. or the major histocompatibly complex antigens over here. And with a peptide over here, these peptides are generated within cells. They are generated intracellularly. These peptides then bind to MHC molecules, and they are presented up here. Once you have a cognate interaction, T cell gets activated, they produce cytokines, they proliferate, and in case they are CD8s, they will kill also. So let's come to the main course. What are antigens? So can you give some examples of antigens if somebody suddenly woke you up in the middle of the night and asked you to name some antigens? What are the ones that you would name? What are some common antigens that you would name?
Bacteria.
Pollen is one, yeah. We have done that. Dust, yeah. Yeah. Any others?
Bacteria, virus.
Yeah, but any specific antigens that you're sort of come across? LPS, lipopolysaccharide, which is often present on gram-negative bacteria. Yeah, that's a good one. Yeah. Any other?
Rely on the bacterial surface, flagella.
Yeah, well, flagella, yeah. LPS, those are all part, the same. Any other ones?
Hen-egg white lysozyme.
Hen-egg white lysozyme, but that's a very, that's more academic, right?
A protein in its native form.
So it's amazing, most of us were vaccinated with a particular antigen, and, it just tells you how short our memories are.
Spike protein.
I'm sorry? Spike protein, yeah, that's what I would have thought, that most people would sort of come up with, one antigen at least that would sort of strike us as the spike protein. Yeah, so anyway, but there are plenty of other examples, right? Antigens are often foreign molecules that are recognized by antibodies or T cells in our body, and the presence of an antigen often triggers an immune response. Okay, so the other ones, of course, are the blood group antigens, right? And it's so important, right? So again, most of us should know, whether you're a A type, or B, AB, or O, right? And if you have these, how is typing done? Very quickly. And imagine if you were transfused with a wrong blood group, what would be a disastrous consequence? It's very important in that sense. So this sort of illustrates why the study of antigens is so very important, right? Now, apart from this, they play important roles in organ transplantations. I will come to this later. So for example, even for typing, for different types of cells, whether they are immune cells, and in this case, the example that I've given is of CD4 positive cells. So if you want, to find out how many CD4 positive cells are in our blood, and that's important because especially if you are following people as they go from HIV positive to AIDS, you need to follow their CD4, CD8 counts, right? And so again, those become antigens. Those become very important for you for us to know, and especially for diagnostic purposes, right? And similarly, if, for example, you have blood cell cancers, for example, knowing what type of cancers it is, there are ways by which we can diagnose. So these, again, become very important. Allergens and, again, vaccines, again, something that we have sort of discussed over here. So are antigens and immunogens the same?
No, sir.
No, sir.
Why not? Spike protein is an antigen as well as an immunogen.
Antigen cannot generate.
Give an example of a difference. In case of spike, it's likely that both would, it would work for both. It would be an antigen as well as an immunogen.
For example, haptens are not antigenic, but they are.
No, they are antigenic, but they are not immunogenic. Right. Yeah. So haptens are an example, right? So we will come to that part. So haptens, for example, they will bind to antibodies, but they are so small that they are not able to elicit. So in fact, if you have this, so the way you get antibody responses to haptens is to conjugate them with larger molecules, proteins, for example. So this is an example that's shown over here. So the hapten is very small, and in this case, an example is DNP, which is dinitrophenol, which is very small, which is, has a molecular weight of 184. It's conjugated to this. So for example, if you want to make antibodies to peptides and all, because again, they would be small, you have to, conjugate them with other larger proteins, for example. And if you inject rabbits, for example, you will have antibodies to the hapten, you will have antibodies to the carrier protein, and you will have some to the mixture and so on, right, which can recognize both. So, these studies, as, was alluded to, Landsteiner is very well known for, his elucidation of blood grouping. But his real, his original work was really done on basis of specificity of antibodies, which was, again, mentioned so well. This is another example. Sometimes you go in the forest and some leaves touches your skin and you come up with something known as dermatitis. So what often happens is some leaves have some oily secretions. And what these do is they bind to your skin proteins. And as a result of which they stimulate your T cells, right? And again, this causes skin inflammation. And, this is very different from the normal type of allergies that we know as. So these are skin dermatitis that is often seen. So again, those are very small molecules, bind to skin proteins, and then elicits this sort of response, right? The other types of allergens, and these are common allergens, pollen, peanut. So, peanut allergy is again very common, especially with, in some kids. So you have to be very careful with it. Seafood is another thing. I have a sort of anecdote over here. We had a student, for example, very smart kid. I remember him. He went with his friends to Goa, and I don't know, usually most of us know if, you're allergic to some things because usually, there is some sort of a thing that you should try and avoid some types of food. Anyway, he went to Goa and he went out with his friends, and he had such a strong allergic reaction that, he unfortunately passed away. And it's a terrible thing, because parents send their kids to study, and the last thing they would expect that the kid to die because of an allergic attack. And they were in a new place. Finding a doctor, hospitals were, was difficult and was really unfortunate. This is real life, right? So this is very important. So what happens in case of allergens or allergies is you have IgEs, and as I said, in the first response, you have IgE being produced, and these IgEs then sit on mast cells, and these mast cells are loaded with histamine-like molecules. So the second time when you come across it, there's cross-linking, and these mast cells are degranulated, as a result of which it leads to, copious amounts of histamine-like molecules being released, which leads to all types of secretions. So when you go to the doctors, it's usually, they're given antihistamines and so on. So this is a striking picture of a mast cell, and you can see one mast cell having so many types of granules loaded with histamine-like molecules. And upon degranulation, you can just see all of them being unloaded. So you can, if this is with one cell, you can imagine, the kind of response that you might get if you have a major one. Okay, so what are some of the characteristics of a good immunogen, right? I think this was, again, came up for discussion. The first important thing is it needs to be foreign, right? So let's say for human CD4 or CD8, take the antigens, but you can make them in mice and so on. But if you wanted, some in mice, you need to, again, move off and make them maybe in hamsters and so on, right? Something very different because rats are still close, and so originally rats were used, but then you were not able to make it in rats, and they moved into hamsters. So those of you who work with mice immunology or with human immunology would know these differences, because you can take a look at the species that the antibody is from, right? So over here, this is an example over here. So this is with a cow or bovine serum albumin. You will not see in cows. You will see a less response in goats, but you will see a big response in chicken because they are the furthest and so on. So then the other one is size. We mentioned haptens, and haptens is a good example for you to know about this. The other is chemical composition and heterogeneity. So here you have, the more complex it is, the more the chances that, our body will see it as foreign and you will generate a good response. But there are other aspects too. So in us, for example, us means humans, you inject us with one antigen and, all of us will sort of respond differently. Why is it that we respond differently? This is because of, again, a locus over here known as the IR genes. IR genes are nothing but basically MHC class II, which is important for T cell responses. And as I said, our antibody responses are also guided by MHC class II, so which becomes important. Other than that, you have different modes, whether it's given IV, IP, intraperitoneal, intravenous, intramuscular, subcutaneous, intradermal. These also are responsible because they might meet different types of cells, as a result of which your immune system may be different. So as I mentioned, with respect to the adaptive immunity, there are two different parts, right? One is specificity. The adaptive response is very specific, right? Whether it's the B cell receptor or the T cell receptor. The second, as I mentioned, it has a memory response. So why is it that we can respond to so many different types of antigens, but not to our own? This was something very important. It was a philosophical question, and you might have seen this during the recent pandemic, for example, right? So none of us had seen, the spike protein or the SARS-CoV-2 virus, not as far as we know at least, but we were able to generate an immune response, right? So I'm sure there would be other types of molecules that, that we could generate an immune response to. So in other words, we have an enormous repertoire to generate an enormous, to generate an immune response. So where is this coming from, right? And so therefore, if we have this enormous capacity to generate an immune response to any possible type of molecule, how is it that we don't generate to our own? And this is by and large, right? Because as we discussed, the immune system is not perfect. You do have autoimmune diseases, but by and large, we are, reasonably okay, because that's why all of us are over here, right? Otherwise, all of us would be sick. So this was a question that a lot of people tried to answer, and a lot of people who've got Nobel Prizes also tried to answer. But the person who actually satisfied this was McFarland Burnett, right, who came up with a clonal selection hypothesis. And you need to appreciate his findings, because this all came about when there was no information on what B cells are, right? What T cells are. In fact, nobody knew of anything about T cells or about the specificities, the B cell receptor, the T cell receptor, and so on. So he was able to come up with some sort of a guidelines for this, and it's only very late that it turned out that his theories were correct. So what he said, and this is very important. What he said is that lymphocytes contain different antigens. So we have an enormous pool, right, of diverse antigen receptors. And the second part of it is that those that bind, right, those receptors that bind our self-antigens, they get eliminated during development, right? So this is, again, something very important. So that is why, by and large, we don't generate autoimmunity, or autoimmunity is not as high as it possibly can be, right? So therefore, those that escape this, those are the ones that go out and seed the periphery. And if they see a particular antigen, they make more clones of that one, right? So you have only clones being made of a particular one. And then, of course, there is more selection. And so this is something very important. So we have an enormous capacity to make different types of receptors, right? And those that see self, those get eliminated. So that's why you have a sort of a safe mechanism. And ones that go out in the periphery, they make clones of their ones. Okay. So this is something, and it's entirely likely that there are many clones that never ever see an antigen in their lives. Only few of them will be seeing, make clones of themselves, and that is what we end up seeing. So that's something very important.
Dr. Nandi,
yeah,
sorry,
yeah. Sure, I will. So this is again very important, and I just want to tell you the importance of this. This is Max Cooper, right? Max Cooper was the one who discovered B cells, and surprisingly, the discovery of B cells hasn't got the Nobel Prize yet, as is the discovery of T cells, that the thymus is responsible for it. But anyway, he got the Lasker at 2019, but a very important thing. So this is what he says. That the clonal selection theory was written with such clarity that one could understand basic principles without deep esoteric knowledge of immunology. Something very important. So I'm coming to the final ones, which is organ transplantation. With respect to organ transplantation, another prominent hero is Peter Medawar. Right? Students should read up on Peter Medawar because he's written extensively, extensive science writer, right? Again, important for students, let me tell you a small story. When Peter Medawar was young, he realized that he was very smart. And, just like you all, right? But he didn't quite know how to channelize his smartness, right? So he asked someone, what do I do, right? So this person told him that you should do something that helps others. So again, something very important for. So at that time, you had the World War II. You had a lot of the RAF pilots coming down, right, and they were getting burnt, right, because of the artillery fire. Right? This is World War II time. So the pressing need was to do something about them, save their lives. So skin transplantations were very important at that time. So this is where he went. He went to a burns unit, and this is what they say about Medawar. It was that he was very precise, and whatever he did properly. He made solid notes of whatever he saw. And he saw that whenever these skin transplants were done, if this transplant was going to be rejected, before the rejection, he saw small cells infiltrating it. And in fact, that was a sign that the transplant was not going to work out. So I remember at that time, they had no idea what these cells were and so on, right? And based on this, he formulated laws of transplantation, where he realized that if you took it from relatives, the chances of, the transplant working were much higher and so on, right? So then, of course, he worked on, acquired tolerance and so on. But, something you all should look up is on Peter Medawar. Subsequently, you had Snell, who worked on congenic, mapped the locus which is responsible for rejection, which is by Snell. Benasraf, I've alluded on the IR genes, which is class 2. And then Dossett, who works on the HLA to try and, in fact, did the initial reagents for mapping HLA were done by Dossett, for which they got the Nobel Prize. So this is the MHC, and again, these are the MHC molecules. This is the MHC class 1, and this is MHC class 2, and they bind these peptides, and these peptides are generated within us. And so this is MHC class 1. If I take a look at the structure, I know it's MHC class 1. And this is from the top. This is the MHC class 1 without the peptide, and this is with the peptide. And to make things easy to remember, it's a vada pav model, right? So you have the pav over here, and the vada is inside, so you can just think of it as a vada pav model. So it's an easy way to remember this. Okay. So with respect to MHC molecules, they are important antigens, but they are important for two aspects, right? Most of us think that they're important because they recognize our antigen, present it to antigens, and you get a response. But before that, they are important in terms of T cell selection, right? So the type of T cells that we get, if you don't have certain MHC molecules, there's nothing to select for, right? So you won't get those T cell receptors, and that's why the diversity that we see is also dependent on our MHC molecules. And remember, MHC molecules are polygenic, polymorphic, and co-dominant. Co-dominant means the MHC molecules you inherit from both your father and mother, right? So that's, again, something important. So I've shown this slide before. I've shown you that this is important, and the peptides are generated from the APC. All right, okay. And again, as a result of which you have your CD4s over here. which are your helpers, and these are your CD8s, which are your killers, which are important. And I'm sure there will be people who will be talking on different subsets of T cells over here. And you need antigen processing and presentation. So there's a difference between antigen processing, because they are processed differently, both MHC1 and MHC2, they're processed differently. I think there might be different people talking about it. I just wanted to inform you. And presentation is important because the presentation occurs on MHC molecules, so MHC class 1 or MHC class 2 molecules. All right, okay, so I am almost done, and these are the highlights. So what I want you to think of is, why we study immunology. Learn from examples, look around your family, friends, be aware of news, and connect. What are antigens? Why is the study of antigens important? I told you about blood cells, blood cell markers, organ transplantations, HLA, allergens, so on. Recall the pandemic in the not too distant past. I can see there's a lot of memory loss of the pandemic. Make sure you stimulate those. Antigens versus immunogens. Are all antigens immunogens? Differences between antigens recognized by B cells versus T cells, clear differences. Why do we not make immune responses to our own antigens? What are some of the tolerance mechanisms? Organ transplantation, MHC, antigen processing, presentation, differences between antigen processing and presentation. All right, so I'd like to thank all my teaching for this, because I teach both UG and PG. And I also like to thank the funding sources and to you all. These are the lab changes during the pandemic. This is January 2020. We were completely unaware of what was going to hit us very soon. This is 2021. We are all masked up. 22 and 24, we are still recovering. This is the main building of the Indian Indian Institute of Science, and this, you can see, this is April because the gulmohars are in full bloom over here. I urge you all to attend the next Immunocon, or the annual meeting of the Indian Immunology Society, which will be in IISc from 17th to 20th October, so please keep that in mind. The last time it was held in IISc was in January 1993, so it's been a long time, so I welcome you all, and thank you all for your attention.
Thank you very much, Dr. Nandi, for a very engaging presentation right from the beginning. We are now open for questions.
It's a loads of information because I am not from immunology background, so learning. So as you mentioned, 2 points I have. So, memory cells, things as you say. So why memory fails sometimes to produce the antibody or immune response, and sometimes it does?
No. So the question is, why is it that some have, some vaccines, you need to give more times because the memory is less, and for some they have, long-lived, right?
Right. But in some cases, some of the vaccine.
Yeah, so smallpox, for example, it's given twice and you have a memory for almost your entire life.
Correct.
Right,
but
whereas for others, you have. So it's just like antibodies also, right?
Okay.
Antibody responses are also, after a point, they sort of wane off, right? So as a result of which you need boosters, right? And with respect to memory, it's the same. Some of the antigens elicit much stronger memory responses, right? So you have some amount of cells that will last for a little longer period. Some you need constant, they need constant reminders to boost it. So that would sort of depend on a variety of factors. The type of antigen, the type of cells it
is that the why booster is given?
Yes, yeah, booster is given primarily to sort of stimulate it, and so get your memory response. I can see the memory response of the pandemic has already died down.
Sure.
Right? So it's the same thing. Right?
And for the sleep time, as you're saying, I know the memory, there's a distinct study known the memory, the immunity is takes a bell-shaped curve during the sleep and then in the morning, and sometimes the antioxidant levels and as well as the cortisol level that varies. So I'm not sure that can be classified as a immunogen, or what is the another one? What is the foreign materials we said that?
Oh, the immunogen versus the antigen.
Yes, it can be called the oxidant has some.
No, it's because we are producing it on our own. So usually they will not generate unless they causes collateral damage or something, right? By which in which case other sort of molecules will be exposed now. Right?
Yeah, but the oxidant and cortisol level is known to, oxidant increase and cortisol level as well as, so is that, I mean, if oxidant means some it coming from mitochondria is also involved.
Well,
and
mitochondria,
stage
or it could be NADPH oxidase
correct,
also.
correct.
Yeah, yeah.
So is that something to, with the immune response?
Yeah, so I mean, some of those are pretty well studied. So the steroids, for example, are known to be known to suppress. Right? In fact, you might have seen that people are given steroid injections, right? And unfortunately, it's an inexpensive method of treating, especially for autoimmune diseases. But steroids have their other effects also. And that is why if you can afford it, some of the biologics like the anti-TNFs and the anti-IL-17s are much better over here, if you can afford it, that is, right? But they are certainly more expensive than the steroids. And steroids would be the first line of
when they're hospitalized and there's a critical treatment.
So that would be known. I was just thinking about, with respect to the oxidative stress, again, that is well characterized. So if you have mutations in NADPH oxidase, for example, you have a disease known as chronic granulomatous disease. Right? And again, that is often seen in kids. It's reasonably common, right? So people have characterized it. So again, if you're not able to generate that, and that's because NADPH oxidase is often present in neutrophils and so on, which are important, again, for protection, right? So if those are not there in your neutrophils and macrophages, then again, you are immunocompromised, as a result of which you have CGD. Which is
the impaired sleep has something to do with immune.
Yes, of course. Yeah, it would be, but natural, right?
Natural.
Yeah.
So one question is that you talked about IR genes.
Yeah, IR genes,
of the MHC class 2 genes.
Yes.
So, sir, what is the role of...
No. Oh, okay. So, the IR genes were discovered by Benacerraf when he found that different mice, for example, just like us, right, given the same antigen, were making different amounts of antibody, right? And then he mapped why this was. And then he mapped it to a part which is the MHC class 2. And at that time, it wasn't clear, because antibodies are made by B cells, where does MHC class 2 come in? And then subsequently it was found out that B cells also need T cell help, which is what I had shown over there. And that T cell help is coming from antigen processing by the MHC class 2. So that's how MHC class 2 is modulating the antibody response. And that's why originally it was found as the IR genes. That's why.
Thank you, sir.
Thank you very much, Dr.
Okay,
Nandi.
thank you.
Thank you, sir. I would request Dr. Sanjeev Kumar to felicitate Dr. Dipankar Nandi. Thank you, sir. We will be proceeding for the lunch. The lunch has been organized in the K block, first floor, and everybody is requested to report back within 45 minutes. Thank you so much.